All right, yeah, Rudy, what you want to get started? I'm good.
Sure, sure. So welcome everyone to our Robert O. Fair Professorship Lecture, which is part of our Natural and Social Sciences lecture series. I would like to acknowledge the Con Edison Corporation, which underwrites the NSS lecture series under the rubric of Science in the Modern World. And this is our fourth lecture in that series this year, this academic year. This particular lecture is actually a little different from the other ones because it is featuring our own colleague, Dr. Allyson Jackson, whom I'll introduce in a moment, who is currently the Robert O. Fair Professor of Environmental Sciences. The Robert O. Fair Professorship is an endowed professorship that we have been able to offer in what used to be natural sciences and is now natural and social sciences for a little over 20 years. The endowment is, we have that endowment thanks to the generosity of the Heinemann Foundation for Research, Education, Charitable, and Scientific Purposes. And if you go to the Heinemann Foundation website, you'll see the full list of kinds of projects that they support. They include a number of things that we would now consider to be under the rubric of sustainability, both environmental, having to do with climate change and kind of toxic environments and cleaning them up, and social sustainability. So I encourage you to go to the Heinemann Foundation website for that. The Robert O. Fair Professorship honors Robert O. Fair, who had passed away in 1998, just before my predecessors applied for this endowment from the Heinemann Foundation. Robert O. Fair was a scientist, an engineer who specialized in sonics, acoustic science. He was a refugee from Nazi Germany who came to the United States and worked as an acoustic engineer for General Electric during World War II. Doing like submarine technology. After the war, he joined together with some other scientists to start an ultrasonics corporation. The Branson, what is now called Branson Instruments, I believe, which is under Emerson Electric. And it was through his work there and in other capacities, also as a professor at Cornell, that he had a life of research in the sciences. His exact connection to environmental sciences per se is not clear to me, but he was clearly a supporter of the natural sciences. He was a longtime member of the Friends of Natural Sciences, who were community colleagues of ours who supported natural science research both financially and otherwise. His wife, Edith Fair, also a scientist, chemist, and biomedical engineer, was also a friend of the natural sciences and was present for many years at this event, at the Robert O. Fair Professorship for many years. She passed away around 9 years ago. So in calling the Robert Fair and Edith Fair to mind, I'm doing so in the spirit with which the Heinemann Foundation supported our efforts to support environmental sciences, and to recall with fondness and appreciation the role that those Friends of Natural Sciences played, along with many others, in making tonight possible. I'd now like to introduce our featured speaker, the Robert O. Fair Professor, Dr. Allyson Jackson, who, as you can see on the website, is an assistant professor in the Environmental Studies program here. Dr. Jackson, I would say that the notes I'm about to read from were composed with the help of Professor Ryan Taylor, the chair of environmental studies, and I am thanking you, Ryan, for that. Dr. Jackson is an accomplished environmental scientist. She has her PhD, she earned her PhD in wildlife science from Oregon State University before joining the Purchase College faculty in 2017. Prior to that, she'd earned her bachelor's degree in biology with a minor in psychology from Juniata College in Pennsylvania and a master's in biology from the College of William and Mary in Virginia. She also worked for a time for the nonprofit Biodiversity Research Initiative in Maine. She's conducted research on a number of different dimensions of wildlife ecology and toxicology, and we'll be hearing about a fraction of that, a slice of that tonight. She's a native New Yorker from Long Island originally, and so after her various studies and work experiences, she returned home to New York State in 2017, joining us in the Natural and Social Sciences at Purchase College. We're incredibly lucky to have her and thrilled to be able to hear some of the results of her research tonight. Allyson.
Thanks, Rudy, and thanks, Ryan. And thanks everyone for being here. I'm seeing a lot of my student names in the list, and it's great to see you all here after hours. So today I'm going to be talking about the Acadia Bug Project, Lessons from a Summer of Science. So first thing is to say thank you, especially to all of the students who were a part of this project. This was really kind of a huge lift on student help for me that summer. Anyone who was up there with us knows that I had a variety of students either there for the whole summer or passing through for part of the summer. So I just wanted to start by acknowledging that all of this data and work couldn't have happened without these wonderful undergraduates who worked with me. So who am I and how did I end up doing research in Acadia National Park? I mean, sometimes I'm kind of amazed that I get to work in such beautiful places like a national park. I am an ecologist. I am kind of an ecotoxicologist almost by accident, just in that I was worried about human effects on wildlife, and pollutants are one of the things that affect wildlife. I'm also an ornithologist, so I really am mostly interested in birds. I am faculty at a primary undergraduate institution here at Purchase College, so we don't have grad students, which is why I think a lot of the undergrads who came and worked with me in 2018 on this project really got a full experience of immersive research in this project. And I think one of the things that I'm learning, especially as I kind of settle into my time here at Purchase, is that I really care that scientists effectively communicate both to other scientists and to the public. My students, we're kind of having a week of presentations this week. So my conservation biology students are doing big presentations tomorrow, where we spend a lot of time talking about how do we communicate our science effectively. And I think that's very, very important in this day and age. So to set up kind of what I've been working on for a long time on mercury, which is a neurotoxin. So, but remember, mercury is an element. It is natural in that way. It comes from rocks. It has that interesting property that it's a liquid metal at room temp. So that has given us a variety of interesting uses over kind of human history, because there's not very many metals that were liquid at room temperature. So it's been historically used and concentrated in weird places by humans before we kind of figured out that it is highly neurotoxic in high doses. So this is the thing that caused Mad Hatter syndrome. It caused a huge amount of developmental issues in Minamata, Japan. And now we know that mercury is a very potent neurotoxin. It causes developmental issues even at low doses. And what a lot of my work over my career has been looking at is effect of mercury on wildlife, and especially birds. So for a long time, I've been looking at this effect of mercury, especially in these particularly contaminated sites. So why do we have a global mercury problem? I mean, part of the kind of story here is that mercury is a point source pollutant, which is where you can kind of picture that, you know, something being dumped directly into the water. It can be through industrial waste. Gold mining uses mercury a lot of times to get the gold out of the ore. We also mined for mercury because we were so interested in using this amazing liquid metal. And so those are kind of places where mercury has been kind of dumped into an area concentrated. That's a point source. But It's also a concern for these non-point source atmospheric movement issues. So coal-fired power plants and other types of incineration and industry will actually release small amounts of mercury into the atmosphere, and then it moves on prevailing wind patterns. So what happens is that this means that mercury can cross basically any border and move into protected areas, and places like the national parks are interested in this because, you know, you can do everything you want within the borders of your national park, but you can't stop the wind from bringing mercury into these places. So thinking about here with our prevailing wind currents, a lot of the kind of air is moving across the country. So anything happening maybe in the center of the country is going to be moved to the East Coast. There's also, you know, like global wind patterns. So kind of this is now, mercury has some localized issues where there's a dump site, but there's also this kind of global movement of it. And it's important to know that mercury is primarily an aquatic contaminant. Some of this isn't that important for you to understand for this particular lecture, but you do need to know that mercury tends to be much more dangerous in aquatic settings. This has to do with its methylation and bioavailability. So in those aquatic settings, it can add a carbon group, it gets methylated, and that's when we can take it up into you know, humans and animals. Not really important that you understand the chemistry of that to understand this talk. Just know that we normally thought about mercury as an aquatic contaminant. And we're also really concerned about the process of biomagnification. So this is that as you move up the food web, you get higher mercury as you move into the higher kind of trophic levels. This is why you often have heard warning pregnant women not to eat certain types of fish, maybe like you don't want to eat a lot of tuna or swordfish. That's because those are low doses potentially of mercury, and with a developing fetus, it can cause impact. So in the marine setting, we, you know, really look and scrutinize those top predators like sharks, tuna, swordfish, that type of stuff, because that's at the top of the food chain and they have the highest biomagnification. So the question you should be asking yourself here is, don't you study little birds that live in the forest? Yeah, I do. I do not study sharks. As much as I like to think that this little guy, this spotted towhee who's clamped onto my hand, is a little predator, they are not what you would think of as a top-level aquatic predator. I am firmly a land lover. I spend all of my time working in the forest, catching these birds. So why is it that I just told you all about mercury in, you know, aquatic settings? So to understand why you need to worry about mercury in aquatic settings, we have to kind of understand an aquatic and terrestrial food web. So here's me on the corner of the water here, looking out over everything. And so if we kind of think about what I'm studying when I'm standing on the corner of an aquatic system, right? I am really interested in these terrestrial vertebrate predators, birds, and I'm interested in what they are eating, because that's where they would get their mercury from, including terrestrial invertebrate predators like a spider and terrestrial invertebrates like caterpillars and other things like that. So you would think that's where me as a forest dweller would kind of live. And then there's kind of this other food web in the water, which is where the mercury is coming from, which is aquatic invertebrates, aquatic invertebrate predators, and then even fish. But the important thing to understand is that we now know that these two food webs are very closely linked, and they're linked through these guys. These are emergent aquatic invertebrates. So these are bugs that start their life stage in the water. So their larval nymph stage is in the water. And then at some point, they go through metamorphosis and become winged creatures that live out in the forest. So there's a whole bunch of these: mayflies, stoneflies, caddisflies. There's also dragonflies and damselflies, which you probably know. All of these started in the aquatic system. So once they're out, they can become food for birds, right? So now we have this much more complicated terrestrial or forest food web where these birds are maybe eating some spiders and some caterpillars, but they could also be eating mayflies and dragonflies. And the important thing that we've learned over the years is that these emergent aquatic invertebrates move aquatic nutrients and contaminants into the terrestrial ecosystem. So on one hand, it's really good. They're moving nutrients, right? Like high energy aquatic nutrients get moved into the forest, but they also can move something like an aquatic contaminant like mercury. So some of my work during my PhD was looking at how in the birds, the amount of emergent aquatic prey in a bird's diet can influence their mercury exposure. So in this graph here, you can see that on the x-axis here, we have proportion of aquatic prey, which means that if you're on this end of the graph, you're eating mostly terrestrial prey. And if you're on this end of the graph, you're eating mostly aquatic prey. And we see this increase with their mercury concentration. So birds that eat more aquatic prey have higher mercury exposure, basically showing, yes, if they get this nutrient from the water, they're also getting kind of a dose of mercury along with it. So I've been working in that for a while. I thought it was really interesting, but I also just think these food webs are interesting kind of on their own. We can study, kind of just how the food webs are set up, and they are especially interesting to me when we even consider kind of human impacts to them. So something that we think about a lot in terms of climate change or even just pollution other than mercury is how it might affect some of these, the ability of these bugs to emerge from the aquatic ecosystem, what that will do in terms of prey sources for the birds. For a lot of our migratory birds, we worry about if the water gets warmer and these bugs come out earlier in the season, but the birds haven't yet gotten here from South America, we start having a mismatch in terms of climate, and a lot of things can go wrong. So it's a really interesting thing to just study on its own outside of the mercury question. And that's kind of my thinking and the idea behind the Acadia bug project. So this project came to life over the years, kind of thinking through with the help of Sarah Nelson and Amanda Clemmer at UMaine, next steps for what we wanted to know about mercury, and also just how we could engage the public. So we kind of had two thoughts. One is that scientists still have a lot to learn about aquatic terrestrial linkages, and the public really doesn't know the importance of invertebrate food webs. I think we think of a lot of emergent aquatic bugs are also mosquitoes or flies or things that we don't like. So sometimes people have a very negative connotation for what bugs are coming out of aquatic water bodies. So the initial project, which was funded through National Park Service, Schoodic Institute, gives out these second century stewardship fellows, which are amazing for early career researchers to come and do research in Acadia National Park. So I was lucky enough to get one of these with my idea to explore Acadia aquatic and terrestrial insect abundance and diversity, but also engage the public in the data collection so that we can get kind of people a little bit more understanding of what's going on in these food webs. So we chose 4 sampling locations in Acadia, and these were chosen because they had already been pretty intensively studied by a much larger dragonfly mercury project. So we already knew a good amount about these sites in terms of their mercury loads. They were also really different, and they were all places where we could get the public to come out and help us. They were fairly safe, not very long walks to get to them, that type of stuff. So the main question from that first SCS funded project was, how do the invertebrate communities differ? And more importantly, if we bring out citizen scientists, do they collect similar data to experts? And this project, all of the students who worked with me were part of this project as time went on, and Matt Garofalo really focused on this for his senior project. When I go talk about it, I'm going to talk about how it also kind of morphed into this collaboration with Lauren DiCarlo at Westfield. So tangentially to that, by the time I was able to go into the field for this, I had already started at Purchase, where I met Dr. Stephen Harris in the biology department, who's doing this really cool work with metabarcoding of different samples to understand what DNA is found in them. So in talking to him, we kind of were like, how can we collaborate? And so we came up with this idea to use what he knew in terms of metabarcoding to figure out what the birds were actually eating in Acadia. And we had 2 excellent students, Alex and Batya, who kind of took this on and used that as their senior project. So that was kind of decided. And then I'm going to talk briefly about what I'm kind of calling my pandemic project, which is looking at what are terrestrial invertebrate predators eating. And this came through funding from the FAIR Award, where I was able to use samples that we had and run stable isotopes on them. So I'm going to walk through these 3 kind of related and complementary projects. So the first question was, how do invertebrate communities differ, and do citizen scientists collect data similar to experts? So in this, we literally were just trying to go out and catch bugs. So we had bugs in the terrestrial world and bugs in the aquatic world. We also did it in two different ways. So we had two habitats and two ways of doing our collection. The first one is what we call expert or quantitative field sampling. This is what the purchase students did while they were out with me. This was very structured. So it was 10 samples of aquatic and terrestrial invertebrates using kind of standard scientific techniques. We did it every week for 8 weeks. It was consistent, thorough, and we sampled only specific areas or quadrats. So if you went to a place and sampled and got no bugs, that counted, right, in this aspect. We were trying to be very systematic in how we're collecting. This is kind of the typical way that you would collect bugs in this area. These were, it's fairly boring, I would say, to do this. I always say if you're doing fieldwork right, it's very boring because you should be doing the same exact thing over and over again. So think about these students who are going to 4 sites every week doing the same 10 sampling over and over again. But then we paired it every other week with citizen science field sampling. So for this, we recruited just anybody. It's a national park, so there's tons of visitors. There's also a strong local community who's really interested in everything going on in the park. So we recruited through the Park Service, through social media, and we said, okay, anyone over the age of 7 is invited. We're gonna teach you all of the techniques, but we did not want people to have to do it in kind of the boring, systematic way that we were doing because it's quite tedious to do it that way. The goal in this was get them out there, get them understanding, looking at these different bugs, how we sample them, and have conversations, engage them, and kind of have fun. So we would bring out these groups to our different sites, and it was a lot of families. We definitely had a whole range of ages over the summer. And we would kind of just say, okay, here's some waders, here's some nets, here's how you do this, go see what you find. And they'd bring us back samples. We did try to say, collect everything. We don't want you to leave anything behind, basically, and we help them work through some of the samples. But I will say this is fairly haphazard sampling in that you can't tell a 7-year-old to systematically check every leaf on a plant. We kind of let them, if they wanted to chase a dragonfly around for a while, we let them. So it was a little bit of chaos, a lot of bit of fun. And so this was kind of our citizen science or qualitative data collection. So the first thing we wanted to look at was the differences in biomass collected between citizen scientists and experts. And this is what Matt worked on for his senior project. And so we were like, okay, well, maybe there's different numbers, amounts of biomass of bugs between these different sites. And biomass can be an indicator of how much food is available for the birds. So using all the data we collected from citizen scientists, we made, graphs, and we could kind of see, oh, there seems to be a lot of variation here. Perhaps Scudic has much higher biomass. But the important thing that we had was we had to compare it to our quantitative kind of accepted scientific methods. And we found that it was quite different what the citizen scientists were able to collect versus what the quantitative collected. So first of all, they definitely collected more. We had more biomass in the citizen science collected samples, but we also didn't even have kind of the same general trend. If you were looking at this top left graph, you would say, oh, this site, Scudic, has the highest biomass, but when we compared it to the quantitative, it's not necessarily showing that it had the highest thing here. So this shows kind of, not surprisingly at all, that haphazard sampling leads to differences in biomass collected, right? We weren't telling them to go sample only certain quadrats. We were just saying, go have fun, collect what you want to. So basically, we can't use citizen science data for understanding biomass. Not really a surprise to anyone. To get a good biomass estimate, you really have to be diligent and calculated in how you're collecting the data. But this kind of led me in also kind of pandemic project related to think through, well, we don't have good biomass estimates, but we do have a good amount of information on what bugs were collected. So when we went out, we'd count up every different bug, and on each day kind of understand that. It looks like chaos in a spreadsheet because there's all of these different bugs that could be collected on different days and different sites. And honestly, it's not something that I had ever worked on before. What it is a community analysis. And I just happened to have a very good friend, Lauren DiCarlo, who is an expert in this type of stuff. So I talked to her and she was willing to collaborate on this. what my understanding of this is that what we're looking at is a biological community that's defined by all the different species that live and interact in an area. If we can understand how the communities are kind of structured, it can help us understand the effective habitat and different food web differences among these sites. And so for this, we could use the abundances that we got instead of the overall biomass, which was kind of a potential way to hopefully deal with some of that error in the citizen science collected data. And so what Lauren did was this community analysis, which is quite complex. It's non-metric multidimensional scaling, or NMS. I'm going to try to explain it as best I can here, but this is why you have collaborators. And it allows us to see if the invertebrate communities differ among the different sites. And more importantly, we can now test if the community is different between collection techniques. So did we actually collect a different community if we were doing this haphazard citizen science, or compared to our quantitative collection? So the first thing that she was able to pull out is looking at indicator species for each site. And these varied among sites and habitats. So for aquatics, we had specific indicators that this is like, these are the hallmarks of this different site, right? So you don't need to know all of these, but this is kind of what we found at each site for the aquatic and what we found for each site for the terrestrial. So we had, at each site, they kind of had different indicator species. The important thing to take away from this list is that some of these are emergent insects and some of them aren't. So here, grayed out, shown in blue, are all of the potential, all of the orders that have emergent aquatic insects in them. So these are the ones that are going to be doing that moving between the aquatic and the terrestrial. It's important because it's going to come up again later that you notice that this site Marshall is the only one in both terrestrial and aquatic that did not have any emergent aquatic insects showing up as indicator taxa. Instead, it had in the aquatic amphipods, bivalves, which are like little clams, and isopods, or aquatic pill bugs, basically. And so those are all kind of indicators of poor aquatic habitat, and then it kind of translated into the terrestrial habitat, where if your aquatic habitat isn't producing emergent aquatic insects, you're not going to find emergent aquatic insects in the terrestrial either. So now we could kind of look at this comparison between our collection techniques. So again, we were trying to see, was the community different between citizen science, that haphazard sampling, and our kind of structured quantitative sampling? And what these look like in terms of community analysis are graphs like this that show overall community structure is kind of bounded by these different polygons here. And so we can see the purple here is our quantitative sampling, and the green is citizen science. So Lauren, again, can explain this way better than me, but the important thing to take away from it is that they don't fully overlap with each other, right? That's kind of indicating that we are seeing slightly different taxa and a community structure in the citizen science collected samples. Again, we were in the same exact spot, so we do not believe that there was actually difference in these communities. If we were doing the same exact sampling, we should have gotten the same answer, but we're seeing some separation in terms of what we were able to collect and what the community looked like based on the collection technique. If you look really carefully at this, it kind of made sense to me, because some of these outliers are like Zygoptera or dragonflies, and we had a lot of little kids out there who just loved catching dragonflies, and you couldn't stop them. It was so much fun. And so it makes a lot of sense that we maybe skewed ourselves towards those charismatic megafauna. Same thing in the terrestrial. A lot of overlap in terms of terrestrial, but some of this kind of non-overlap in terms of what the citizen scientists were collecting. So these NMS plots are showing differences in community structure based on the collection technique. Definitely showing that these citizen scientists, either in the way that they're doing it or how we kind of gave them free rein, are more likely to focus on certain taxa more than others. I mean, not surprising at all. In some ways, you would almost be surprised that there was this much overlap for the amount of people and chaos and things that were going on those days. I was actually pleasantly surprised. I felt like we were getting a pretty good representative sample when we had all the families and things out with us. So conclusions about this part. Citizen scientists definitely in this aspect are not going to collect exactly the same data as our expert sampling. But we definitely felt like we achieved our education and outreach goals. We got a ton of people out collecting bugs. We got little kids out who said, I don't want to touch a bug. And then they were all of a sudden in waders grabbing mud and getting dragonfly larvae out of it. So from a scientist perspective, it's kind of like, well, so it's not perfect data, but that engagement might be really, really important to me. And that might be something that I care about. And I really wanted to look at, well, maybe they are still useful though. So the thing that we got was a lot of biomass because there were a lot of people out. We had a lot of hands kind of helping collect data. And so that's when I started thinking, well, maybe we could use that high biomass that we collected for future stable isotope analysis. So using what they collected and sending it out for an analysis. So this was my pandemic project. I had kind of started this collaboration with Dr. Christina Murphy up at UMaine, where theoretically we were going to go into the field last summer, and then COVID kind of stopped that. So as you know, I was looking ahead to not having field work to do, I started thinking, well, what could I do, especially because I had a junior leave sabbatical lined up right then. And so I thought through, we have all these samples. What about if we ran stable isotope analysis on them? So I wanted to see, can we use stable isotopes to understand the differences in aquatic and terrestrial food sources? So stable isotopes, again, same as mercury, like there's some stuff that you're not going to understand in this very brief lecture. The important thing is that it's basically the carbon or the nitrogen fingerprint that's related to where the actual molecules in your cells came from. And it can be used as a tracer to understand who is eating who. So when you eat something, some of the carbon and nitrogen from that thing is incorporated into your cells. So here we were now looking at our invertebrate food web because that's what we had these good samples of. But you have to think about stable isotopes really give you a very coarse estimate. They kind of are like, in general, this is terrestrial carbon and this is aquatic carbon. It's not going to give us exactly, you know, that this dragonfly ate a mayfly that day. So what the stable isotopes kind of look like are plots like this, where we can look at the differences in kind of prey that's available. So in this case, we have terrestrial prey kind of in the bottom right and aquatic prey in the top left. And then what we can do is look at predators and figure out, well, is the predator eating more aquatic based or more terrestrial based? So if we had something like a spider who's just a little predator, right? They can either be eating fully terrestrial prey or maybe they're eating fully aquatic prey. And that's what we can use these stable isotopes to kind of understand. So I pulled out a bunch of the kind of prey sources, ran them for stable isotopes, and we also ran a bunch of the predators that were out there. So the main predators that we had were dragonflies and damselflies, daddy longlegs, and spiders. So it's important to know that these guys are different. So the dragonflies and damselflies started their larval stage in the aquatic system. They actually came from the water. And so we'd really expect them to have a lot of aquatic carbon in them. They went through metamorphosis, but the interesting thing is once they're out into the land, they're predators. So they can go out and eat, you know, spiders and caterpillars. They can eat whatever they want once they're out in the terrestrial land. So when we look at this, this is the same plot as before, but now we have predators mapped onto it. We can say, okay, if they could either eat aquatic or terrestrial prey, they eat, you know, these based on their stable isotopes. So if we look here, these are 2 dragonfly samples that we had, and we can see that somewhat surprisingly, it seems like they are kind of mapping close to terrestrial prey, which means that they have been out of the water long enough and eating, and they end up eating a lot of terrestrial prey. We can also see that most of the spiders at this site were eating primarily terrestrial prey, except for this guy up here. And I was kind of perplexed by this until I remembered that we caught an actual spider like living in the water. Like it was like on reeds and stuff in the water. And so that was obviously somehow getting more aquatic prey that day. So for the most part, these spiders are eating terrestrially, and then we'll have some outliers who ate super aquatic at this site. And we can do this over and over for all the different sites and look at what was available for them to eat and what they ended up eating. Here at Schoodic, we see kind of most of our spiders are mapped over terrestrial, and then our dragonflies are kind of coming closer into the aquatic side. I feel like this whole seminar is going to be Allyson and the people she called during COVID for help. This one was Dr. Matt Ramirez, who helped me figure out how to do this kind of complex Bayesian analysis called mix-SciR, so that we could look at the proportion of aquatic or terrestrial carbon in these different predators. So when we looked at the different predators and then looked at the variation in the actual aquatic carbon that they were eating, we can see a few kind of trends showing up. One of which is spiders on the left here varied a lot. Some spiders were eating an awful lot of aquatic carbon and some were eating very little, especially at certain sites. So this red site is Gilmore Marsh. We had a lot of variation there. We had a mostly low in some of the other sites, except for, again, this is that one marsh spider that we caught. Dragonflies and damselflies. So I am surprised here that they had such a high terrestrial reliance. Again, these came from the water. You would think they would have a lot of their carbon still devoted to aquatic-based carbon, but we saw at a couple sites like Marshall and Schoodic, they were actually showing a good amount of terrestrial carbon signature. And then for the most part, all of the daddy longlegs, the Apiliones, were eating mostly terrestrial prey. So this led me, this is another part of it. So we looked at kind of the really coarse scale stable isotopes that were giving us generally what carbon these organisms were eating. But then I also was working with Stephen Harris to look at what the birds are actually eating. So now we're kind of adding birds into the food web again, looking at what is coming out of them. And so again, this whole project was in Harris's lab with Batya and Alex as seniors. In 2019, they graduated. And so what we did in the field was when we catch birds, which I catch them to be able to get a small blood sample for mercury, we would collect a poop sample, a fecal sample. And this is pretty easy for birds. They poop a lot, and we have to kind of hold them in these cloth bags that you can see in these photos. It's a way to keep them from being stressed. If they're out in the open, like if you had them in a cage or just holding them, they can get super stressed. When they're in a closed-in bag, they can stay warm, dry, and relatively unstressed. The nice thing is when they're in that bag, they often poop. And so we would then collect that poop in the field and preserve the DNA. So here you can see a Moor who is charged one day with scraping all the poop off of the bags and putting it into preservative. And then we could bring those back to the lab for analysis of DNA sequences. Again, this is something that was kind of handed off to everyone in the Harris lab, and they troubleshot how to do this very complex metabarcoding stuff. These students were amazing in not giving up. We had a lot of false starts in terms of Stephen had never done bird poo before. I don't know a lot of these techniques, and Batya and Alex did a really amazing job in sticking with it and getting some interesting results. So metabarcoding gives us a super fine scale analysis. If stable isotopes is coarse, this is like finer than fine because it knows the actual species in a lot of cases, because now we're looking at the actual DNA that passed through the bird, is in their poop, and we can look at species level differences. This is just an excerpt of the type of table that we would find. So some of them, it depends on what's available in databases, but for a lot of these, we can get down to genus and species. So we know that that bird was eating this particular Diptera species. This is super helpful for species like the Diptera or the true flies, where some of the species in this order have terrestrial origins and some of them have aquatic origins. So now I can show you in gray all of the times that a bird was potentially eating a fly that came from the aquatic system. So we can get a lot more fine scale analysis here. And so what we looked at was at each site, what were the birds generally eating? So this is the percent of birds that these different orders showed up in. So here again, we're looking at Marshall Brook. And the black bars are aquatic based and the gray bars are terrestrial based. So the 5 birds that we caught at Marshall all had terrestrial beetles in their diets. And then we can kind of go through and look at kind of less common. And we could do this over and over again for all the different sites and kind of understand variation in what was kind of the most used prey item in these different sites based on what the birds were actually eating. And so what I really was curious about was, okay, so we can know exactly what they're eating. We know what that bug was. Can we get an idea of how much they're relying on aquatic prey and use that to understand their mercury exposure potentially? So we were able to calculate something called this metagenomic operational, whatever, you don't have to worry about that. Percent aquatic MOTUs are the amount of aquatic species divided by the total number of species in an individual bird's fecal sample. So if we got 10 DNA sequences in there and 5 of them were aquatic, it had 50% aquatic. And so we saw a lot of variation when we did this calculation in the amount of emergent aquatic prey found in the fecal samples. So again, we see, Gilmore here on the left has this, really wide variation in the amount of aquatic MOTUs found in it. And so I wanted to look at, for each of these birds, if you were a bird that ate 100% aquatic, does that mean you also have higher mercury? So I looked at the comparison of those to mercury and really found very little correlation. So the number of species that they were eating of aquatic didn't seem to predict their mercury exposure at all. And I think what's going on here is, though we get this really fine scale resolution, we also have a very fine scale time resolution here. What we're analyzing when we analyze one poop sample is whatever happened to pass through the bird at that exact time. And they can kind of, what their mercury is looking at is an overwhelming, like the last few months, what they were exposed to. So I think it makes sense that what they ate that day before, which is what our poop sample, isn't tied to the overarching amount of mercury that they're getting dosed with. So I'm not really surprised by this, but I also just think some of the information that we found just in general about how much aquatic prey they were eating is really interesting in and of itself. So what did we end up learning about mercury in birds from all of this? Again, I spent a lot of time in the mud and working with bugs than birds for this whole project. So we were able to get blood samples from some of the birds and run them for mercury. That's how I created that graph on the last slide. And so if we look at overall mercury in birds among the sites, it seems as though Marshall site keeps showing this low kind of mercury signature. If you remember back, Marshall was also the site that had no emergent aquatic indicator species in Lauren's analysis. And so it starts to highlight maybe something's going on with this particular site. And so I did a really coarse analysis, and this, will not stand up to peer review because it's 4 points. But if I looked at the average number of emergent aquatic insects in the sample that we collected versus the average bird mercury, we are starting to see these three sites kind of cluster in terms of, medium mercury and medium and high emergent insects. And then the Marshall site is kind of down here doing its own thing. Marshall site was chosen because it was a pretty degraded and nastier site than the others. It's going to go through a bunch of restoration, so we thought that that was an interesting thing to study. And so it was cool to kind of have it kind of show up as an outlier. The hard thing for me with all of this, which is, a science communication issue, is that it's important to remember that these nutrients are very important. So I'm talking about it in terms of, oh, the emergent aquatic insects are giving mercury, but really they're also providing a really important nutrient. The same thing happens a lot of times when we talk about mercury in fish. Fish and their omega-3s and all the good things that fish provide, you don't want people to take away, never eat fish because there could be mercury in it. It's more like weighing the consequences of these different things. And that's what we need to think about for birds here, is that in general, I would rather see an ecosystem with a good amount of emergent insect activity, because that kind of highlights that it is a healthy ecosystem. So overall conclusions, yes, citizen scientists collect slightly different data, but it can still be super useful. In my case, I used it for stable isotopes, which can help us understand who is eating who at a really coarse scale. And I found that spiders and dragonflies eat varying amounts of aquatic prey. There's a lot more to kind of dive into in that study. We found that fecal metabarcoding reveals who is eating who at a fine scale, and that the birds in these areas are definitely eating emergent aquatic insects. And that, even though we didn't find the correlation to mercury, it makes sense that maybe there could be some movement of mercury into the birds through those emergent aquatic insects. So the future work that's coming down the pipe is actually doing what we meant to do last year, hopefully getting out to Acadia, working with Chrissy at UMaine and USGS to kind of fill in some of these gaps from the first year. The awesome thing is that I'm going to get to go back and catch birds and fill in what I really wanted to know in terms of stable isotopes and birds. And Chrissy is a fish biologist, so she's going to kind of fill in the food web on the fish side. So I can keep my expertise on, land and forest, and we are all going to learn a lot from Chrissy and how she samples fish and learn a lot more about that side of the ecosystem. And I think all of this is really interesting, even without the mercury component, just because how all of this is set up is just such an interesting puzzle to try to unravel. So I have a dog here who's getting very excited, but I'd like to acknowledge a whole bunch of people. I'm going to leave this up so that you can look it over. There were so many people that ended up helping with this, including all the students that I thanked on the first slide, all of the citizen scientists who came out to kind of help. This one in particular is important to me. That's my godson, Philip, who made it up to Maine because we're doing this. And I just think this whole project was really cool in that way. It's a way that I could share some of my science with people who, you know, don't necessarily normally care about what I'm doing or can come out into the field with me. So it was a really great experience. So with that, I will take any questions though. Are there any questions? I can see the chat if you want to put it in there, or you can also unmute and ask me questions. Oh, buddy's here to help. Oh, Sarah. Oh, hi, Sarah. How long after emergence does it take for isotopes ratio and the prey themselves to shift from aquatic to terrestrial? Yeah, so the interesting thing for a lot of the like mayflies and stoneflies are that they don't even feed when they're out in the terrestrial. So like I should have said that, like that's an interesting thing that we can look at those where they don't have the chance to incorporate terrestrial carbon into their signatures. The dragonflies, I haven't looked into that. It must be short based on this. Like, I mean, those dragonflies only live, I think, a few months, up to maybe 6 months, or before they migrate. So I'm curious to see how quickly that kind of turns over in them. Because yeah, that having them show that terrestrial signal could mean that they've been out for a long time when we sampled them and they were getting lots of terrestrial prey in that amount of time. Oh, Dan. Hi, Dan. We found very few spiders being eaten by the birds in general. Yes. I mean, we had really low sample size for that meta barcoding part of the project. I think there's more to be seen there. I think Acadia is kind of different than some of the other places that I've worked in that some of the sites had just relatively low prey in general. So I don't know. It could be also why, you know, the birds in Acadia don't have super high mercury. It might have to do with that they aren't eating a lot of spiders. I think that's something that we can get into more this summer when we do kind of more stable isotope sampling and to try to understand, like, we can really focus on the spiders and try to figure out like what's going on there. Because yeah, I mean, your work is definitely showing how important those spiders are. Catherine, hi. Is there any relationship with proximity to salt water? Very good question. No, they were not all the same inland. And I've wondered that about Hunter. When we were at Hunter, we were very close to the bay and to the salt water. And I often wondered that in terms of the birds that we caught had relatively the same mercury as some of the other sites. And I wondered if they were getting any aquatic marine derived nutrients from that. I think if I had to do it again, you know, maybe I wouldn't use that site. It was the only one that was like super close to the ocean. But other than that, it's definitely something to think about. We're missing that part of kind of the food web for sure, if it's contributing to anything. And I should say this summer we're going to different sites, so we won't be able to like, we're gonna, you know, try to go to new places instead of redo these analyses. So I think we've maybe created, the stable isotope part of it has definitely created more questions than answers in some ways, but I'm still really happy that I was able to do that, especially when, you know, it was COVID and we couldn't do anything, because I think it gives us a really good idea of what we're up against in what we're doing now, which is a much bigger stable isotope project.
Allyson, I had I wanted to, first I wanted to add some acknowledgements to that lovely acknowledgements page, which is that here in the School of Natural and Social Sciences, just putting this show together today, Harrison McDonald and Amanda Zalo played very crucial roles in getting us here virtually. And Gord Caruth is always helping getting, processing payments and procurement of many things, and also helped me learn the history of the FAIR professorship. So I want to acknowledge his help with that. I have a very kind of simple question coming from my non-natural science background, which had to do with the citizen science work that you talked about, which I found really interesting. And I really appreciate the way you talked about it, as well as seeing those great photos. But I wondered, you said that there was, I think you used that great expression of charismatic.
Charismatic megafauna, yeah.
Charismatic megafauna. I'm going to try to weave that into a conversation of mine in the next week or so. But I wondered if also, again, I feel like it's a very simple question, but are some easier to catch than others? Like is it not just a question of what cool things I'd rather catch, but also that just some things are just more elusive than others?
Well, yeah, and dragonflies should have been more elusive. That's the thing. Like, it's hard to catch a dragonfly, and they were definitely chasing them down because they wanted to see them. And we were kind of in the view of they're learning a lot about dragonflies right now. And then once we caught one, we could talk about it. Those shouldn't have shown up predominantly, especially in the terrestrial setting, for sure, because they are hard to catch. But yeah, I think there's definitely, and there could be some avoidance of some, like a spider. So there could be some ones that are just too ewy to pick up, and that could be part of it. What we saw more often was potentially just ignoring really small things because they're not, you don't even necessarily notice them in a net if it's really, really small. Whereas when we were doing it, we were keyed into like every single thing that was caught had to be processed. When we have all these kids out there, they're kind of picking the biggest creepy crawly and putting it in the jars. So I think, that could definitely sway that data a good amount, and all of that stable isotope should be kind of taken with a grain of salt because of that. But in stable isotope studies, a lot of times you are looking for kind of the dominant things there, so it's not as important that we get that perfect like differences in biomass or anything like that. I think we were just doing PR for spiders all summer, telling people how cool they were. So in the future, we could do a full spider study in that way and, have the bravest come out and we'd catch spiders.
Yeah, look forward to that. Could even engage our colleague in psychology, Paul Siegel, who's dealt with arachnophobia. Right?
Yeah, I did not realize that was a thing until I've been doing so much work where we catch insects. And then I have to admit that some people are very, very scared in ways that I am not scared of spiders because I'll just pick them up with my hands. Great. I mean, it was wonderful. Thank you all for coming to this. Like, good to see so many students. I hope I didn't disappoint you all since I've been preaching about science communication and good presentation styles all semester. Any other questions?
Getting lots of praise and appreciation in the chat, and I would like to echo that vocally. Thank you, Allyson, for all this incredible work and for sharing it with us and doing it in such an engaging fashion. Really made our evening very educational and enjoyable. Thank you.
Yeah, thank you, everybody.
Thanks.
See everybody tomorrow for our cons bio presentations.
Good night, everybody.
Bye.